Good afternoon, everyone. Welcome to the third day of the EDQC. We had a very interesting discussion for the two present days, and I'm pretty sure that you will be very active today to ask a lot of questions to our two keynote speakers. Today, it's going to be a very micro-focused in the presentation. It's my pleasure to introduce you the process and product quality focus we will have today with a presentation by Professor Paul Cotter on the application of microbial research on food production and processing, and also a presentation from Dr. Xiao on the impact of spore-forming spoilers on the quality and shelf life of dairy products, which is a very important issue for the quality of the dairy products worldwide. Paul is the head of the food science at Chagask, and he's a renowned microbiologist in Ireland, but globally worldwide. It's always a pleasure to enjoy his lecture. He's also working on a spin-off in Ireland, which is not mentioned in his bio. I'm afraid it's difficult to have the bio of Paul in one slide. So what you need to know is that it's always a pleasure to be lectured by him, and he's always very insightful in his questions. So really looking forward to today's presentation. I was told that you cannot click on the link. That's the issue indeed. But if you go on LinkedIn and you search the keyword European Dairy Quality Conference, it's very easy to find. And with that, I'm going to hand over to Paul, who will present you the topic of omics.
Super. Thank you for the very nice introduction.
Welcome.
Thank you for the introduction. So I will share my slides now. So can you see that well? No?
Yes.
Okay. So good afternoon, everybody. It's really a pleasure to be here today to tell you something about our research relating to relating to the study of food chain and foods using microbiome research. And I guess we're applying this sort of technology right throughout the food chain, not only to look at foods, but the food processing environment. And that extends to the dairy chain, but also various other different food environments as well. Okay, so whoops. Just bear with me a moment. Great. So I'm going to be focusing on culture-independent approaches to microbiology research. The reason being that there has been a revolution, I guess, in this sort of research over the last decade or two, whereby increasingly there's an appreciation that traditional culture-based approaches don't really allow us to see all of the complete picture. And in some instances, we're really only just seeing the tip of the iceberg. And the reason for that is that agar plates and broths and such like are only really allow us to grow a small proportion of the microbes that are present in any one environment. And so even in the best case scenario where we use lots of different agars and lots of different temperatures and lots of different pHs and hundreds of different variables, you still will only be capable of growing a percentage of the microbes that are present in any one environment. And then you might only find out what's relevant with regards to bacteria, not yet necessarily the fungal population or the viruses or phage and so on. So because no ideal agar or ideal culture approach is available, you're only ever really going to see a small proportion of the entire picture, and yet unless you use another strategy. And so our strategy is to use culture-independent approaches rather than culture-based approaches to see what exactly is happening. And so in this regard, metagenomic approaches, where we're securing all of the DNA from an environment provides us with this complete picture that we seek to find out. And so I guess using the analogy that has been used in recent days of the going on a bear hunt, well, I guess you could regard culture-based approaches as allowing you to go on a hunt for a particular type of a bear, or perhaps a bear and a snake. Whereas the culture-independent approaches, which are, I suppose, bacteria or microorganism independent or non-specific, would allow you to look for a bear, a snake, a shark, a whale, a mushroom, and everything else all at the one time, ideally. We're a while away from getting exactly there, but we're making significant progress across the research field. So the initial premise relies on extracting DNA from the material, or RNA, and I won't get into that right now, but I can address it in the questions later on. But after you extract the nucleic acid, then there's a number of different approaches that you can take, and those fall generally into one of two different categories. One being amplicon sequencing, which is one whereby you pick a particular target gene that is common among the microorganisms that you want to focus. So for example, 16S sequencing is used to look at bacterial populations. And so that allows you to amplify the region using a PCR approach that will bind to the flanking conserved regions of the gene. And the variable regions within that gene can then be compared with a database to tell you what microorganisms are present in your dairy powder, in your food sample, and so on and so forth. Now, the issue with amplicon sequencing is that you need to know which type of microorganism you want to target initially. In that, do you want to focus on bacteria and use 16S sequencing, or fungi and use ITS sequencing? And the other issue is that it will tell you, it will just give you an identification down to a genus level. So it will tell you within your sample that you have Bacillus present and Clostridium and Lactobacillus, but it won't tell you if it's Bacillus subtilis or Bacillus cereus in the vast majority of cases. The alternative approach is one that we refer to as whole metagenomic shotgun sequencing, or shotgun sequencing. And in this instance, rather than sequencing a particular gene that's present or common among bacteria or yeast, in this instance, you take all of the nucleic acid material and you sequence it in a random fashion. So all of the bacteria, all of the viruses, all of the phage, all of the small eukaryotes, etc. And in this instance, as well as getting you a community composition insight, which will get you to species or even strain level, you also get further insights in that you can see what sort of genetic pathways are present. In other words, how many genes associated with antimicrobial resistance, how many genes associated with virulence are present. And even indeed, following on from Adriana's presentation yesterday, you can begin to assemble some genomes from your metagenomic DNA. So you, in this instance, rather than culturing individual isolates and doing a genomic analysis on these individually, you can instead take all of the vast amount of information that you have and then put that into different pots representing the different genomes that you have present. Now, there are technical challenges relating to all of that again, we can maybe get into in the question and answer session later. So unsurprisingly, with the revolution that these new technologies have brought about, they have been applied in a vast variety of different ways with respect to foods and the food chain. And while I'm going to focus on the food chain specifically today, I will draw your attention to the fact that this approach has been used extensively for different foods. In our group, we focus primarily on fermented foods, and these are just individual examples of very many that we could refer to. So quite often people look at taxonomy and other different pathways that are encoded. And in addition to looking at individual foods, we've recently also published a paper that just came out last week, where we've also looked at an even broader, wider variety of more unusual fermented foods, including things like different kvases and boza and different types of kimchi and wagashi and things like that, to really compare and contrast the microbiomes present in different foods. So this may appear initially like a bit of a stamp collecting exercise, but it actually does have translational value in that you can try to identify spoilage microorganisms and pathogens, look at flavor compounds and how individual microorganisms correlate with flavor, with a view to trying to develop new generations of food with particular flavors or other specific attributes. And obviously you can delve down into a lot of fundamental science, as we've done with this recent study relating to the cheese microbiome. In this, we looked at 55 new newly sequenced artisanal cheeses from across Ireland, but combined that with 129 microbiome samples from other cheeses across the world, and really did a very in-depth analysis of what's present and what new different species we can identify, looking at relationships between phage and CRISPR and phage resistance and so on and so forth. So I'll draw your attention to that, but I won't refer to it in any greater detail today. What I will do is continue to focus on the food chain microbiome and our evolution in the approaches that we've taken to looking at this over the last few years. So one study that we found particularly interesting at the time is one that was published in 2017, where we looked at different samples collected from a farming environment over the course of the year, both during winter and summer, and during a period when the animals were outdoors versus when they were indoors. So this is 16S data, so the kind of the older approach that's just focused on bacteria. And this may look a little bit like abstract art, but I'll explain just exactly what we're looking at in a moment. So all of the symbols up here, so this is a beta diversity plot, and each symbol, each circle or a triangle represents all of the vast amount of thousands of microbiology sequence or microbial sequences that are condensed down into one data point. And if 2 points are located quite close to one another, it means that the microbiomes are quite similar. And if the 2 points are very far apart, it means the microbiomes are quite different. So you will see from the legend here, so that all of the circles represent raw milk, in this case from the indoor season, and the circles down here are raw milk from the outdoor season. And all of the other triangles represent different environmental samples. So we're seeing environmental samples during the indoor season from silage, from the bedding, from the teat surface, from indoor feces. And you can begin to see that the teat surface samples are those which are located most closely with the raw milk microbiomes and therefore have the most in common and are the most likely source of potential contaminants. During the outdoor season, you can again see a similar phenomenon. So the circles here are the raw milk. Outdoor feces seems to be a bigger driver here, with teat surface being slightly further away, with grass and soil being of lesser importance. So the same can be applied within the processing facility. And we discussed yesterday, or you would have heard Adriana talking about the importance of new genomic approaches and accreditation of those approaches with a view to the rolling out of them and making them more widely available. And obviously, metagenomic approaches are even further away from getting to that point, but they are of use as a tool in order to give you a deeper insight. And in this case, rather than looking for a pathogen, in which case you would require accreditation, we use the approach to identify a spoilage microorganism with quite a degree of success. So we use this approach to look at a phenomenon known as pink discoloration defect, which a number of you might be familiar with. This particularly affects certain types of cheeses, particularly continental type cheeses across the world, and is a phenomenon that has been was first noted in the scientific literature about 100 years ago. So we were approached by a PhD student who worked with a colleague of ours in Teagasc in Moorpark, co-supervised by Dermot Sheehan. And he had endeavoured, after very many others had tried over the last 100 years, to determine the microbial basis for this phenomenon or to determine what else might be going on. But lo and behold, as for very many others, conventional approaches didn't reveal any difference between the microbiology of control and defect cheeses. So we applied our metagenomic approaches using a number of different control samples versus defect samples. And you can see immediately that all of the defect samples have something in common in that they have this extra lilac or purple color here representing a particular type of microorganism referred to as thermus. And when we looked at thermus more closely, we realized that the specific species was Thermus thermophilus. And this attracted our attention because Thermus thermophilus is a heat-loving microorganism that you don't typically look for in a food environment, but it is found in particular environmental locations around the world, like for example in Yellowstone National Park, where it's associated with hot thermal springs and the amazing colors that you find in those springs as a consequence of the production of carotenoids. So that made us think, could perhaps the presence of thermos be resulting in the production of a carotenoid that could be contributing to this phenomenon? So I don't get into this in great detail here, but to prove this whole concept, having established that thermos was present, we used specific challenging culture-based approaches to grow thermos successfully from these defect cheeses, and then reintroduced thermos into new cheeses at the beginning of a production cycle, and recreated the defect problem, therefore establishing the thermos was indeed responsible. And we've gone further to look at the carotenoid that is being produced and to identify different control compounds to determine what exactly is going on there. Ultimately, we also collected a lot of environmental samples from the processing plant and established that the particular issue seems to be traced back to hot water systems that are used for the production process. So based on those sort of insights, we've decided to take an even greater look at some of the processes that are involved in the production of various different dairy products. And so I'm going to focus for the remainder of the presentation on two different examples that were led by a PhD student, now a postdoc of ours, Aoife McHugh, who did some fantastic work in this area. So the first study that Aoife did related to the production of some skim milk powder. And what we did was we collected milk from the bulk tank of multiple different farms, 67 different farms, and analyzed all of those individually. So all of those milks were transported in 11 tankers to a production facility. So we sampled from each of those. And then we collected samples right throughout the process, from the whole milk silo, pasteurizer, and right along the way, and from the ultimate powder that was produced, to look to see how the microbiology changed at each step, and did any microbes transfer right the way through, or if other microbes ended up in the final product, where were they introduced during the food process. And then we did that on two different occasions during the year. One during the mid-lactation period, when the cows in Ireland are still outdoors, and during late lactation when the cows are indoors. Again, at first glance, this may seem like abstract art, so don't worry, I'll explain it in more detail. So this initially is our 16S data, or data which will tell us the names of the bacteria, but just at genus level. And what you're seeing as you go from left to right is the bulk tank, the tankers, whole milk silo, skim milk silo, right the way through until you get to the final powder. And we have some information relating to the cream as well, but I'm really going to focus on the skim and the final skim product and the milk that we began with. So you will see here that what we're finding during the late lactation period is that the milk that comes in is dominated by Pseudomonas and Acinetobacter, representing the cold chain storage of the milk. And these microbes linger and continue on through the process despite pasteurization and all of those heat treatment steps. And they're still the dominant microbes in the powder at the end of the process. Now, these microbes were also the dominant microbes during the early mid-lactation part of the study. But in contrast, in the final powders, those microbes, the Pseudomonas and the Acinetobacter, disappeared, and instead you see a dominance of spore formers, Geobacillus and Thermus, which we trace back to arising during the production process, and we attribute them primarily to the biofilms that we believe formed along the way, and that survived the CIP process. Now, we're not saying necessarily that this is representative of what happens in mid-lactation and late lactation constantly. These are just two snapshots in time, but it would be intriguing to extend this analysis out over multiple days to see if this is a consistent pattern, and therefore are there different precautions that you need to take in recognition of different times of the year, or might it be down to just different meteorological conditions or some other factors that drive the differences that are resulting in your final milk powder. We also carried out the more in-depth shotgun analysis and found a similar type of outcome. So again, from the control, from the tankers right the way through the process to the skim milk powder. And during mid-lactation, we're getting confirmation that Thermus and Geobacillus dominate. The beauty with the shotgun data is we can go further and we can tell you which particular species were present which particular geobacillus and which thermus. And similarly with Pseudomonas, we can see in black here that it continues to dominate in late lactation, and we can tell you that it's Pseudomonas fluorescens is that which is present. You can also do lots more with the data to identify, as I say, virulence factors and other things that are present, but I won't linger on that for today. So by way of summary, from this first example, I just wanted to reiterate that obviously dairy microbiota changes through the milk powder manufacturing process, and you don't need very high powered and very detailed microbiome analysis to tell you that. Again, something that you knew already, but there was an early dominance of cyclotolerant spoilage-associated bacteria. We see potential proliferation of low levels of thermophilic bacteria present in raw ingredients, or their presence within biofilms within the processing plant, seemingly result in their final domination at certain other times of the year. And we would propose that the routine implementation of high throughput sequencing methods can lead to an understanding of the reasons underlying the dominance of different microbes at a time. So the beauty being that you can look at multiple different microorganisms in combination rather than just using one agar for each of those independently and then having to do a subsequent deeper analysis to characterize those. Okay, so a second example is one which is we've put on bioRxiv as a preprint. It's in revision at the moment, and we expect that it will be accepted for publication within the next month or so. So we're giving you an early preview on this. And that's really an example where Aoife took her previous study and tried to take it to the next level, whereby we focused more extensively on shotgun analysis alone, but also try to look to see to what extent we can translate this approach, which is the previous approach is one, I guess, that you could use in an academic lab and isn't so practical from a real world scenario, to try to make it more applicable to actual food processing facilities. And the reason why I say it can be more applicable is that the logic is that instead of using these large, very expensive sequencers, such as are generated by Illumina, who are the market leaders in DNA sequencing, that instead one can use devices such as this Oxford nanopore technology device, which is known as a MinION, which is about the size of a USB key. And so is handheld, can be used in real time, and you connect it up with a laptop to do your analysis. And so could these sort of devices be used to try to get a better real-time understanding of the microbiome of a food facility? Now, there are obviously challenges. One of the issue relating to the technology that's required for running these sort of instruments is that you need more starting DNA or more material in order to do your analysis. So if you're doing a swab of taking a swab from an area that has just been cleaned, where there's a very low microbial load, then it's going to be challenging to get enough DNA to do your microbiome analysis. But I'm going to describe in a moment some of the ways we got around this problem. So before proceeding, we just had to make sure the instrument could, in theory, successfully discriminate between different microorganisms of relevance to the food chain. So we set it the challenge in that we created a mock community of microorganisms, picked some spore formers, Bacillus cereus and Thuringiensis, picking both of those particularly because they are so closely related that it can be very often difficult to differentiate between the two. And then two other spore formers, Bacillus licheniformis and Geobacillus staurotermophilus. And so we use both a 16S approach using this instrument and also a whole metagenome shotgun sequencing approach on the right. And what you can see here is that the 16S approach works reasonably well. It still is challenging to differentiate between the Thuringiensis and cereus, so I wouldn't be fully confident in ascribing the species level here. The whole metagenome sequencing also identified all of the target microorganisms at the species level, and you see them again at the genus level. It also incorrectly picked up a Bacillus paralicheniformis, but that amount was so small that it's not even visible on the graph here. So quite a degree of success, and that provided us with the confidence to proceed. As mentioned earlier, you can also use that information to compare or to assemble genomes or metagenomes, metagenomic assembled genomes, and this all worked out very nicely as well in that we identified a number of different plasmids, we identified our 4 key microorganisms that we introduced into the system successfully. So it all looked very promising. So we then went to a processing facility and looked at a number of different environmental locations to see how well we could identify what microorganisms were present. So I'm going to ask you to maybe pay a little bit more attention in particular to the next section of this slide, because it sets us up for what I'm going to talk about over the course of the next few slides. And so what we did is we extracted DNA. And so for some of that DNA, we just sequenced it as normal on an Illumina sequencer with no special processing. So that's NextSeq, no pre-processing. I mentioned earlier the MinION requires more DNA for analysis. So we use an approach whereby we randomly amplified the DNA we extracted from these environments to get more of it to facilitate sequencing. So this is MinION, and it was referred to as the multiple displacement amplification is the means that we use to get more DNA. So this is MinION MDA. As a direct comparator, we wanted to use this same approach, but with MDA as well, in case MDA introduced a confounding effect or a bias. And so we did that again for Illumina. And then finally, we wanted to see how culturing could impact and to what extent culturing biases the result. So we plated out the material onto an agar plate, and then we took all of the material from that agar plate and sequenced it all using next generation sequencing on our Illumina approach. And finally, also plated on a variety of different agars, but you just use more conventional 16S sequencing of individual colonies to look at all the various different outputs. So again, this may initially give you a headache if you try to interpret what's going on, but I'll walk you through it slowly over the next few slides. So we had samples that were collected from the same locations in October, November, and December. We have different locations from the door, from a table, from an evaporator, and so on and so forth along the end. I don't expect you to pay too much attention to all of those. And then we have the amplification, MDA for MinION, MDA for NextSeq, the no pre-processing for NextSeq, and the plating approach before doing any sequencing. So we included our mock community that I mentioned previously, the 3 different bacilli and the 1 geobacillus, to make sure everything was working nicely and we could have faith in our results. And all of the different approaches successfully did this, so we were very pleased with that. Next, we determined the impact of pre-processing and the sequencing type. And so if you compare the same sample and using different approaches immediately down onto one another, you see in some cases that they're very similar, in others less so. The observation seems to be that there are certain taxonomic groups that provided us with a bit more of a challenge. Cocoria, Acinetobacter, Enterococcus, and the Pseudochromobacter were some of those that varied. In particular, the samples that had low microbial load to begin with were the more challenging, and those with a high microbial load, we see quite a high level of consistency, regardless of the approach used. So we're quite pleased with that. In contrast, though, if you compare these images with the ones above, you will see quite a bit of difference when we first plated out the microbes onto the agar. In other words, we're selecting just for those microbes that can grow on the agar, and all of the other microbes that don't like that agar or are difficult to grow aren't properly reflected. So it's a much less diverse combination of different microorganisms represented here. If you take that to the next level, and we actually, instead of using next generation sequencing at all, but use conventional sequencing of colonies, so the way in which we present the data is a bit different. But here we use total plate count agar, and then we selected for thermophiles and skim milk agar. You see sometimes you've only 1 or 2 colors representing how few microorganisms grow. And sometimes we didn't detect any microorganisms at all. A little bit more variety from the TBCs, but it's still the identity of the microbes is quite different from that which you would see from the more in-depth sequencing-based analysis. So while there are challenges relating to this sort of approach in terms of sourcing sufficient template DNA, and there are some consistencies when you use different sequencing approaches or platforms, and also we need to improve the databases to better identify microbes present, especially those that aren't easily cultured. I think there is, from these results, undoubtedly considerable merit in proceeding towards looking to see if we can apply them on a wider scale, maybe on a monitoring or a non-official or accredited basis initially to help processors to identify problems and help to resolve them before they occur. before eventually moving towards accreditation at a later date. And so we're now trying to apply this approach on a broader scale and to push it forward to make it even more translated and to address some of those issues that have arisen or could potentially block their further development. And we're doing that through an EU project that I'm fortunate enough to coordinate, known as MASTER. And so MASTER is a large project that spans a number of different institutes across Europe with 30 different partners, and it's 2 years into a 4-year project now. And we're looking for all sorts of different applications of microbiome research to the food chain, probiotics, diagnostics, and others. But one of the work packages particularly relates to databases, pipelines, DNA extraction tools, the use of handheld sequencers, and so on and so forth. And that work package is led by Nicola Sagata and Chris Creevy. And what we're doing there is developing SOPs for validated sample collection and storage, and different validated approaches for 16S and shotgun sequencing, addressing the absence of certain microbial representatives in metagenomic databases, so that when eventually the hope is that when a processor or a quality assurance person in a plant has their handheld DNA sequencer, then they can send that data to the cloud and have a quick analysis that will give them a very rapid insight into what's present in their sample without the need for a dedicated, experienced bioinformatician. Already we've optimized our swabbing approaches in terms of the type of swabs and the extraction approaches that are optimized. And we're carrying out shotgun sequencing of 100 different processing plants from across all of the partners across Europe. A final mention, Francois kindly mentioned a spin-out company that I've been involved with that I spend maybe 1 day a week working on, which is Seek Biome, and we're looking at applying this and other sorts of technologies to the food chain, but also to human health. And so obviously if anybody is interested, I'd be happy to talk about that in the future. For now, I'd like to acknowledge all of the various different collaborators and people within our team that have contributed in various different ways over the recent years, including all of the different master team and our project manager there. And finally, to thank all of you for your attention. It's very much appreciated. Thank you.
Thank you, Paul. As a pastoral microbiologist, I must say this is pretty disturbing that there are more to see in a food that you can see on a agar plate. But it's good because I always been very careful when people mentioning that you have a standard for total plate count, and I say, whoa, I'd like to see this. I am not so sure total plate count really exists. Speaking of difficult cultural organisms, spores are pretty complicated. There was recently a very interesting work done by NISO, and this time not on dairy but on cocoa, on the different spores. We have a working group from the ISO TC35 and C9 on developing methods for spores. Maybe we'll get there someday. And it's a very vast domain, and there is a lot to see about this. Going to the last slide. So we come to the famous, now famous Q&A session of the day. Thank you everyone for attending. As you see, there is, as for the previous day, a strong link between the two presentations. We see from what you explained, Dr. Shaw, that spore formers are pretty vast as type of microorganisms. There are a lot of different species, which mean that culture-based methods will have their limits because you cannot find bacilli and clostridii on the same agar media. Also, the growth potential is very specific, whether it's a requirement for absence of oxygen or specific nutrients. In that regard, we often see a misfit between the microbial specification, which refer to total plate counting, and the real spoilage in UHT milk, for example. Do you believe that at some point it might be interesting, both of you, to have a routine NGS techniques in order to more tackle the spoilage issue?
Can I just give very quick answer? And my idea is from case we have seen the spoilage case for the UHT products. Most cases they are not mixed culture; they're very pure culture for specific case. So I think, and they're normally they're culturable. Only one case I have to say they're not, because that bacteria can only grow in the product. So when we put it out there, they're not cultivable. So I think for that could be meaningful. But for most of the cases, as far as it's pure and cultivable, I think maybe it's overkill to do the routine analysis.
Thank you, Dr. Shaw. Paul, what are your view on this? Do you believe routine NGS is an utopia or something that could come in the laboratories in the coming years?
Yeah, so I think there's merit in rolling out these technologies more widely. I guess as long as we rely on traditional approaches, we're only going to see the traditional answers. And so there may be instances where, kind of like the cheese pinking scenario, we see a microbe that's doing something that for now we don't realize has a microbial basis. And I do agree with Chow that in an instance where you're have clear spoilage that's likely due to one particular microorganism, that culturing can give you an answer. But you also quite frequently have an instance where, for example, a powder doesn't give you a problem until it's mixed with something else, and it's then that other microbes begin to flourish and to contribute to its spoilage problem. So to have a better sense as to what's there and what they might be capable of doing is useful. Ultimately, I think having extra knowledge is never harmful. It just comes down to a cost-benefit decision then.
Yeah, extra knowledge is always better, but it's better if you're able to do something out of it. We have been discussing in the previous day that being proactive will be, for example, looking in the environment. Here, as mentioned by both of you, is also looking at different point of the process chain in order to better get understanding of what's happening. One interesting point, or maybe challenging point with type of spoilers, you were mentioning Clostridium tyrobutyricum, that it often happens in the silage issue for hard type cheese. So how do you, as a factory, leave the factory in order to anticipate the problems? Do you think that there will be benefit to use these new techniques, which are not fully understood yet by the food industry in primary production, or is it a bit premature to go on the fields?
So I can maybe answer initially
Yeah,
in that.
both of you. Yeah, Paul, please.
So I guess, again, I would agree that there is merit. I think a lot will come down to how quickly these newer handheld devices and similar approaches become more widely available, but also cost effective. So ultimately, finance, our budgets, our economy will dictate a lot of these things. If ultimately you could get to a scenario whereby the milk producers and milk processors can come up with an incentive, such as they currently give for the quality of raw milk relating to the number of the number of somatic cells present, or TBCs or something like that. If you could envisage a scenario in the future where there is some other incentive whereby, in the absence of undesirable spore formers or something else in your milk, it would be of greater value, then you would imagine that milk producers will become very much more interested in providing those results then. Again, but it will come down to the cost effectiveness of the assay.
From my point of view, I think stands on ALA, we are dairy cooperative, which means we will collect the milk from the farmer, so almost unconditionally, which means we need to be very careful about what kind of method to be used for routine analysis, or what kind of method to be used for the troubleshooting analysis. So that's actually first thing I'm talking about NGS. In my personal opinion, I think so far what we understand about metagenome, particularly shotgun, the cost still a little high, and then we probably more in the short term will use the for troubleshooting method to illustrate what kind of what group of bacteria has been involved. Which are actually the real causative agent of this spoilage. And then when we turn to the routine analysis or routine method, we probably will think about cheaper one and easier to perform, or maybe faster techniques.
Yeah, you reach a very sensitive point, Xiao. If you want to use a routine test for sorting the milk, if we take an analogy to the antibiotic residue testing, it means you need to have all the methods with the results available within 2 minutes, because that's the time allowed when you receive the tanks and you know where you're going to divert. So routine testing for the time being might not be the best option. If you need to wait even 2 or 3 hours for each tank entering the facility, that's not acceptable. Having said that, could you maybe consider an option at the farm, at the tank level, by cistern on every factory, of routing the milk depending on the Clostridia population or the Bacillus population, whether it will more be fit for UHT, for R-type cheese? I know it's not available yet, but is it a type of solution that could be of industrial benefit?
That's a very great question. So we have been a few running external or open innovation projects. So we are expecting some kind of early warning system. So when we have the silo or when we have the tank milk, they can, by some fast or quick method, within 2 hours, we will know how to use this tank.
Okay. One of the sensitive points with testing is that it always comes to compliance, non-compliance. Presently within IDF, the Standing Committee on Harmonization of Microbial Methods is working on developing an ISO standard for the count of Clostridia spores, specifically Tyrobutyricum and Butyricum, which is used as payment to the milk producer. Right now, everything is culture-based, and we know there are a lot of limitations. How could we use the insights of NGS, as shown by Paul, in order to develop more targeted culture-based techniques for the milk producer? And I know this might look two different worlds, but there is one, as you both of you mentioned, is providing a lot of information, cannot be used in routine, and there's another one which is, yes, limited, but cost efficient and can be used in routine. So how do you make the communication between the science generated by NGS and the application on culture-based media?
So I would suggest in that regard that the two can be very compatible, in that the more you know about the microorganism that you want to target, the better you can make those assays. So if, for example, for the Clostridium butyricum or Tyrobutyricum, it's possible to look at all of the genomes that have been sequenced from representatives of that species, and also some others from metagenomic assembled genomes. And from that, identify the unique pathways and innate antibiotic resistance and other things that are conserved among those particular strains and species that are absent in others, to try to develop better agar methods using carbohydrates that only they can use, or combinations of nitrogen sources, and so on. And so that's happened in the gut microbiome space, whereby previously uncultured microbes can now be cultured because it was possible to get their DNA from fecal samples and figure out how those microbes could grow. And I envisage that it might be possible to make better culture-based assays using the same sort of premise.
Yeah, I think from my point of view, I think I have a two angle. For African, metagenomics, like say, or barcode metagenomics. I think that's a perfect tool for the troubleshooting and the spoilage investigation as a microbiologist in dairy. And then for the shotgun metagenomics, it will be very promising to use it to get out from framework of taxonomy. I can give you a case here. Powder quality was challenged by nitrite content. And then we have knowledge, and then I think it's our colleague in New Zealand has been done a lot of study on that, associated to the thermophilic bacteria growth in the evaporator before the spray drying. And then also turned out this behavior is not for specific species. Actually, it's very heterogeneous for the specific strain or specific subgroup or subpopulation. So in that way, also not necessary to be geobacillus, can be other bacteria who has high expression or production of the reduced enzymes. So for this kind of subject, which not necessarily to be linked to specific species or genus, I think we can use this shotgun metagenomics to look for the specific target or biomarker, introduce functional genomics into this concept. And then we have a method to detect a certain biomarker related to this enzyme activity. And then we don't even limit everything to specific genus or specific species. That would be my wish for the future connection between the metagenomics and the, let's say, spoilage control. I hope that makes sense.
Yeah, it makes sense. I try to think about the next question. I need to be careful because this will implicate standardization. And so, Adrian, okay, you can jump in if you want. One of the issue we have with ISO TC 35 SC9, so the Technical Committee Food Subcommittee Microbiology, is that most of the methods are culture-based. And presently, IDF, on the initiative of Chagask, I should say Ireland, with Mark Fenelon, will be working for dairy science and technology on implementation of all these new techniques on a routine basis by the food business operators. How can we try to standardize and get recognition by every stakeholders of this type of techniques? Because if you change the techniques, for example, for Clostridium tyrobutyricum for the payment of quality of milk, and maybe at the end of the day you're more accurate, but maybe this accuracy can be something a bit tricky for the primary producer. Do we need at some point standardization? Is publication enough? How can we get also regulatory recognition of these new techniques?
I would say it's not too dissimilar to the development of any or introduction of any new technique, which in the past might have included the introduction of a new type of agar. And so rigorous testing would have to be performed across multiple different labs to ensure a consistency of results and engagement with industry and all of the different stakeholders to ensure there's buy-in rather than trying to enforce a new approach that isn't agreeable and may meet resistance from others. So I expect it would require the involvement of a direct comparison between culture-based approach and some of these newer technologies across multiple labs with, for example, spike samples to ensure that the outcomes are realistic, to introduce live microorganisms, introduce dead microorganisms to ensure differentiation can occur. I didn't get into it during my presentation, but we did use steps during our approach to remove the DNA from dead bacteria. But all of those things need to be done to provide confidence before pushing forward.
Adrian, your mic is on, Adrian, if you want to join the conversation.
Yep, can you hear me?
Yeah, we can hear you.
Okay, perfect. And I think it's an interesting approach. The first problem we have, and you raised it also, and Zhao raised it, is the specifications. And I think any new, radically new method, it has to be reflected in the specifications. And at the moment, what we're trying to do in the ISO working group, and I fully agree, it's very difficult to have a total plate count. And this is why we rejected the notion of a total spore count, because there is no such thing. You will never be able to cultivate in one method all the spores. So what we're trying to do is to do a risk profiling to see which of the spores, spore formers are important for which of the food categories, and then to try and harmonize the method as much as possible to really be able to cultivate the main spore formers. And if you miss one, for example, Bacillus sporothermodurans, you will miss it for UHT milk because it needs such a specific method. This then needs to be highlighted in the ISO method. So I think if we want to make any big change in the method, it needs to start really with the specifications, knowing what to measure, what is important, and then the methodology can follow.
Before you speak, Tosha, to complement the point of Adrian, by definition, by the Codex Alimentarius, microbiological criterion, it's the scope, and there is a method. So the point is with these new techniques, as you mentioned, Adrian, and also Xiao mentioned, the more you investigate, the more you see that the scope is more than anticipated. And at some points, sorry to be a bit down to earth, we need to have boundaries, because at some point, sorry, it's I'm French, I'm a bit pragmatic, but it's payment at the end. So how do we make all this approach? And we see that it needs also training from the authorities, recognition by the regulations, the science being accurate, the industrial having a tools fit for purpose. It's a global set.
Yeah,
Xiao,
and a lot needs to change.
But Francois, I think for me, the life-death differentiation is still quite important, because in Europe, we are still arbitrary to perform pasteurization. So, I mean, using metagenomics for the raw milk, I mean, I remember there was a study done at the UC Davis, or I think 2 years ago, but we got a lot of reads from metagenomics on the vegetative cells, but we know they are going to be killed by pasteurization anyway. So what's the meaning to sequence those?
I will give you an example, not per se the raw milk, but there is a huge trend to use aromatized UHT milk, and one of the most appreciated flavor is cocoa, chocolate milk. The issue is we know for many years that cocoa is full of spores, and as long as you've got your cocoa powder, or if you got your chocolate, which is a delight, by the way, hello to the Swiss, Belgium, and the French, but if you put in the milk, this is where the issue begins. So you were mentioning, Xiao, that you should have a look at the raw material, the ingredients, and this is why I was mentioning initially this work from NISO, because NISO is mostly oriented with milk, but they work for the cocoa, because there are bacilli everywhere, but not the same bacilli. So how do you put a limit with this? I think we're lagging the show. I don't know, Adrian or Paul, if you want to. Yeah, Asha, where have you? Yeah.
The proper heating.
Troubleshooting, yeah. Yeah, but troubleshooting is after. How do you try to be proactive and, for example, make sure which cocoa powder could be fit for chocolate milk and which should not? I mean, is it something that the dairy industry must do? Is it something that the cocoa industry must do? Is it something that both industry must do? I'm not saying this because I worked for the cocoa before, but it's a lot of knowledge on different microbiota from different food matrices.
I think for cacao, the cacao itself doesn't contain spores. The spores come during the cocoa powder producing process, during the fermentation, during the drying, during the grounding process, as far as I know. And then I think some proper control process by the cacao powder supplier, still the most efficient way to control the thermophilic spores there, to improve or looking for the source of contamination of the spore-forming bacteria. But there's not so much what dairy company can do, because we, as you said, we mix the non-dairy ingredients to the milk and then heat treat it.
Yeah.
So
Thank you. Yeah, please.
can I maybe add also, I mean, there are other instances too where you have, for example, thermostable enzymes that are produced by a spoilage microbe like a Pseudomonas. So while the Pseudomonas might be killed, you still would want to know that a Pseudomonas was present in your raw materials, because that will linger through the process and cause problems later on there. And I guess in terms of rolling out these sort of approaches, I would again imagine that they would be most attractive in situations where there is most money to be made. In that, if you compare a dairy commodity such as skim milk powder, maybe there isn't enough of a margin there, but perhaps the first port of call may be, for example, the infant milk formula industry, where there are specific microbes that you can't have present due to the safety, but also because the product is of greater value. And so maybe that might be the industry that will be pushing back those boundaries initially. These routines are not so uncommon in clinical diagnostics and in hospital labs, so they are out there. It's just a case, but it's always been the case that all of the approaches that have been introduced there happen there maybe 10 or 15 years before they become routine in the food industry.
Yeah, definitely. So thank you, Paul. Thank you, Xiao, for your excellent presentation today and for this insightful discussion with a guest invite, Adrian. And I will leave the floor to Rob for the concluding word of this third edition of EDQC.
Yeah, thank you very much indeed. So just a few more seconds, everybody, in conclusion, before we close out this year's Dairy Quality Conference 2020. I'd like to express on behalf of all of the bioMérieux organizational committee that's been involved in putting this together, a big and sincere thanks to each and every one of you. Thank you to everybody that has connected across the 3 days and indeed today. I'm pleased to say we had more than 500 connections across those 3 days. So I'm hoping that's a testament to the quality of the content as provided by our esteemed speakers. So with that in mind, I would like to also, of course, thank all of the speakers across the 3 days. So to Maria, to Christophe, to Jack and Adrienne in the previous days, and indeed here now today, Paul and Xiao, thank you very much indeed for what I would consider to be very relevant, very insightful and engaging presentations. And I believe that our audience would agree with us on that, considering the number of people that we have had connected. Final word of thanks indeed to our intrepid moderator, Monsieur Baudéchamp. Thank you very much indeed. Very well and deftly you steered the ship all the way through to straight into port, and I would say more or less without any major casualties. So well done indeed. And please, everybody, we've mentioned it several times, but don't hesitate to take advantage of all of the replays that are available on the LinkedIn channel. And if you haven't signed up to that LinkedIn channel, please do. The community would welcome you, and we'd love to keep the conversation going in the coming weeks and months. Dairy problems have not all been resolved over the last 3 days, I'm sorry to say. So let's keep that discussion alive and well. And I look forward to seeing you all hopefully in person for next year's EDQC 21. Thank you, everybody, indeed. Have a good and fantastic rest of the day. Cheers.
Have a good afternoon. Goodbye, everyone.
Bye. Bye.